Now let me explain you how the enzymes restriction endonucleases ligase helps. Say for example, this is the organism, this is the oh no, let me show you like it's more simplified. Okay, say for example, this is the DNA, and this part of the DNA we need it. We use restriction endonucleases and it cuts the DNA. There you go, the part of a DNA that we need has been cut. Now say for example, this is a plasmid. Let's see now. Yeah, so this is a plasmid, circular DNA. We use restriction endonucleases, the same restriction endonuclease, so that the size of the cut is same. Say for example, we have cut this part of the plasmid, and now the plasmid is like this, because the other part which we have cut is here. Now this part of the DNA that we need will be inserted into the plasmid. Okay, so yeah, it was green color, so yeah, so we have inserted it here. Now they need to form bonds. So how do we form bond? We will use the enzyme ligase enzyme. The ligase enzyme will connect them, and thus the plasmid will look like this. So what I want to say is that in the new plasmid, this was the old plasmid, in the new plasmid, this part of the gene is needed to be carried to the organism that we are going to modify. When this plasmid has been taken to the organism, that if the cell has accepted the plasmid, then the cell will have this gene, and the cell can produce the protein that we need. Okay, so tools for gene technologist. So a gene technologist needs a set of tools for genetic engineering. Okay, first we will know what is a restriction enzyme. I gave you a small idea. Now let's see it in details. Restriction endonucleases are a class of enzymes from bacteria which recognize and break down DNA of invading viruses known as bacteriophages. So restriction endonucleases actually a class of enzyme. There are many types of restriction endonucleases. Like there are many types of carbohydrates like amylase, maltase. Okay. So this is actually a group of enzyme, a class of enzymes which is present in bacteria. They use it to break the DNA of viruses which may destroy them or harm them. If they break the DNA of viruses, the viruses cannot multiply inside the bacteria. We will know how bacteria and virus causes infection in our chapter of immunity and also infectious diseases. We have also had a brief idea in our O levels. Now, bacteria makes enzymes that cut phase DNA into smaller pieces. These enzymes cut the sugar phosphate backbone of the DNA at specific places within the molecule. Specific spaces. Okay, and sugar phosphate backbone, it means that the phosphate is linked to the ribose sugar of the next DNA. It cuts that bond. This is why they are known as endonucleases, which means within. They work within the nucleus or within the virus or cell to cut the DNA. Their role in bacteria is to restrict a viral infection, hence the name restriction endonuclease or restriction enzyme. Each restriction enzyme binds to a specific target site on the DNA and cuts that site. Bacterial DNA is protected from such an attack either by chemical markers or by not having the target sites. Now, since the enzymes are in bacteria, it may also cut the bacterial DNA and cause it harm, but it does not happen. That's because I told you that restriction endonucleases are specific. They need a target site. That is uh, the like the target site. I means the the markers of a cell, the chemical markers of a cell, like the glycoproteins which are present on every cells. Each cell have an antigen which is its chemical marker. The bacterial cell do not have that antigen, but the viral cell or virus have that antigen. Or by not having the target site, or by having a chemical marker that is a specific antigen which may recognizes the bacterial cell as its own. These target sites or restriction sites are specific sequence of bases. So the target sites are specific sequence of bases and the chemical markers are antigens. For example, the restriction enzyme called BAMHI always cuts DNA where there is GGATCC sequence on the strand and its complementary sequence CTAGG. Obviously, guanine is complementary to guanine is complementary to cytosine, adenine is complementary to thymine. You'll notice that this sequence is same in both direction, like GATCC, and that's this one. If you read it in a opposite direction, GGATCC, it is palindrome. If this type of sequence is present, then it is called palindrome, and like palindromic. So many but not all restriction sites are palindromic. Many but not all restriction sites are palindromic. Okay, this is an information for you. Restriction enzymes either cut across the sugar phosphate backbone or give it a blunt end, or they cut it in a to give it a blunt end, or cut it to give it a staggered fashion to give it sticky ends. Now, say for example, let me show you. If say this is a sugar phosphate backbone, these are the bases, and if that restriction enzyme cut it like this from say this part, from say this part, it's going to have like this. So it's going to have like this, and there will be the other part. Okay, like this, the other part. So what will happen is that there will be, say for example, one nucleotide cytosine, and there will be like guanine, and there will be like adenine. It could be anything. And there will be like thymine. So complete set of DNA here and complete set of like there is not that other complementary strand can bind to it, but if it has sticky ends, if it has sticky ends, it's going to look like this. There will be not one strand absent around this part and another strand there. So complementary strand to this guanine, adenine, thymine and cytosine can bind to it, and so it is called sticky because complementary stranded DNA will bind to it. But if it is cut like blunt across, then other DNA cannot bind to it. The sticky ends are short lengths of unpaired bases. They are known as sticky ends because they can easily form hydrogen bonds with complementary sequences. of bases on other pieces of DNA cut with the same restriction enzyme. It has to be the same restriction enzyme so that the strands are complementary. When long pieces of DNA are cut with a restriction enzyme, there will be a mixture of different lengths. Obviously, to find the specific piece of DNA requires involving separating the lengths of DNA using gel electrophoresis and using gene probes. These techniques are described on page number 470 to 471. We will come across these techniques, but for now just understand that for the separation and identification of our wanted of the gene that we want, gene of our want, we need to identify them using the process of gel electrophoresis and then using gene probes. These techniques are described. Okay, multiple copies of the required piece of DNA can be made using the polymerase chain reaction. How the polymerase chain reaction occurs, it will also be described. I told you that polymerase chain reaction is used to make multiple copies of a single DNA. Well, gel electrophoresis separates the DNA according to their size, and gene probes are like the complementary DNA to the part of the DNA sequence of DNA we need, and those parts will form complementary, and then we can again separate it to get the DNA we want. It is a short, like how it works in a short term. I just told you. We will look at it in detail. Let's see this one. Restriction enzyme ECORI. Its restriction site is this one. Now, site of cut across DNA. It cuts it like this one. So from guanine, its complementary is cytosine. In adenine, its complementary is thymine. So it gives it a sticky end. It does not cut it across. Okay, look. When it will be cut, this thymine, adenine of the lower will form complementary sequence with the adenine, thymine sequence of the other DNA. Same goes for this part. So it will be, a sticky cut, sticky end. It will have sticky ends. But here, it will have a blunt end because they are already paired with their complementary nucleotide base. And so they won't form any bonds with other DNA. Okay. So this enzyme HAEL number 2 cuts the restriction site, this sequence, in a blunt way. Now there are also the sources of enzyme. You do not have to learn by heart any of this. This is just for your understanding, because when you will be working, this data will be given to you. Okay, so restriction enzymes are named by an abbreviation which indicates their origin. Roman numbers are added to distinguish between different enzymes from the same source. If, say, example, from one bacteria, different restriction endonucleases have been restriction enzymes have been identified, then Roman numbers 1, 2, 3 are used. For example, ECORI comes from this bacteria, this certain strain of coli, and was the first to be identified from this source. Okay, so this is the strain RY13. Now that many proteins have been sequenced, it is possible to use genetic code to synthesize DNA artificially from nucleotides rather than cutting it from out of chromosomal DNA, or making it by reverse transcription. Now, since many proteins has been sequenced, the codons of the proteins have been identified. For the advancement of our tool, we can select those nucleotides with the bases and make them join into their into the sequence we want and make the DNA of our choice. We do not have to make it by reverse transcription or cutting it out of the chromosomal DNA using the restriction enzymes. Genes and even complete genomes can be made directly from DNA nucleotides without the need for template DNA. Because before, the part of the DNA, I mean the sequence of the DNA which you needed, had to be had to be cut by an restriction enzymes, and then separated by gel electrophoresis, and then identified gene probes, and then cloned by PCR, polymerase chain reaction machine, and then we would get the gene or the sequence of DNA we want. But now we can simply make it, using the technology. So we do not need template DNA. Scientists can do this by choosing codons for amino acid sequence they need. The sequence of nucleotide is held in a computer that directs the synthesis of short fragments of DNA. These fragments are then joined together to make a longer sequence of nucleotides that can be inserted into plasmids for use in genetic engineering. Plasmid is a vector. We will know what is a vector later. This method is used to generate novel genes that are used, for example, in the synthesis of vaccines, and they have even been used to produce the genomes of bacteria consisting of millions of base pairs. Now, what is a genome? We have come across this term for quite a lot of times. Genome is the complete sequence of nucleotides or the genes in an organism. Okay. Okay, so vectors. As I've explained how a vector is used, or I've explained like liposomes and some other vectors. Okay, so let me get into details. Getting gene into a plasmid vector. In order to get a new gene into a recipient cell, that is the cell in which we want to put the gene or introduce the gene, a go-between called a vector often is used. The gene cannot be directly taken into the DNA of the cell. It has to be taken in by anything, and that anything is called a vector. Now let us talk about a plasmid vector. One type of vector is plasmid. These are small circular pieces of double-stranded DNA. Plasmids occur naturally in bacteria and often contain genes for antibiotic resistance. They can be exchanged between bacteria, even between different species of bacteria. These are information learned by heart this. If a genetic engineer inserts a piece of DNA into a plasmid, then the plasmid can be used to take the DNA into a bacterial cell. To get the plasmid, the bacteria containing them are treated with enzymes to break down their cell walls because the plasmids are inside the bacteria. The naked bacteria are then spun at high speed in a centrifuge so that relatively large bacterial chromosomes are separated from the much smaller plasmids. I have explained the physics behind this in the chapter of nucleic acids and protein synthesis. The circular DNA of the plasmids is cut open using a restriction enzyme. The same enzyme as one used to cut open the gene should be used so that the sticky ends are complementary. Like if I use the restriction enzyme, each restriction enzyme is actually a group of enzymes. So each restriction enzyme has its restriction sites where the enzyme cuts the DNA. So if I cut the DNA at a specific part of the DNA that you want to put in, we must use the same restriction enzyme so that the complementary strand in the plasmid can be cut. So that after that, when you will put the gene and mix the gene with the plasmids, they can be complementary. Okay. If a restriction enzyme is used that gives blunt ends, the sticky ends needed to be attached to both gene and the plasmid DNA. Because if the restriction enzyme gave a blunt end, some restriction enzymes gives blunt end like this one, HAEL number 2, blunt ends, then sticky ends need to be attached artificially to both the enzyme and the plasmid DNA, because also the same restriction enzyme was used to cut the plasmid DNA, and so that plasmid DNA will also contain blunt ends. the opened plasmids and the length of DNA are mixed together. Some of the plasmid sticky ends pair up with the sticky ends of the new gene. The enzyme DNA ligase is used to link the sugar phosphate backbones of the DNA molecule and the plasmid, producing a closed circle of double-stranded contained DNA containing a new gene. So the plasmid, when we cut it using, I have explained how restriction enzymes work. Okay, I have explained it. So this is now recombinant DNA. Bacterial plasmids can be modified to produce good vectors. Okay, plasmids can also be made artificially. Yeah, so for example, the PUC group of plasmids have a low molecular mass, so they can be readily taken up by bacteria. An origin of replication, so they can be copied. Several single target sites for different restriction enzymes in a short length of DNA called a polylinker. So suppose a plasmid have only one restriction site. So only one restriction enzyme can be used to cut the part of the plasmid, and that restriction enzyme will be used to cut the part of the gene, and the gene will be inserted into the plasmid. So only one new gene can be introduced. But if it has like a polylinker, that is different restriction enzymes, single target sites for different restriction enzymes, then different restriction enzymes can act and cut different parts of the plasmid, so that those different restriction enzymes can be used again to cut many different types of genes, and so the different types of genes can be inserted into the plasmid. So more than one type of gene can be introduced into the cell using just one single plasmid. One or more marker genes allowing identification of cells that have taken up the plasmids. So the new plasmid have one or more marker genes allowing the identification of cells taken up by the plasmid. So it just has a marker gene attached to it. So if it has a marker gene attached to it, then we do not have to use artificial or extra genetic markers. Okay, so you will understand how a genetic marker works later. Plasmids are not the only type of vector that can be used. Viruses can also be used as vectors. A third group of vectors are liposomes, which are tiny pairs of phospholipid containing the DNA. There are more about these other vectors later in the chapter. Okay, so let's see the picture. What happens? This is the bacteria. So that we can get the plasmid. It is treated with the restriction enzyme to cut this part, and this is the gene which you want, the green part, the human gene. So it is also treated with the same restriction enzyme, so that the sticky ends is complementary to the sticky ends of the plasmid DNA. To the plasmids. So then the DNA and the plasmids are then joined together. They form complementary base pairing. They are treated with DNA ligase to form the bonds, and so this is the recombinant plasmid. Okay, with the gene added. This green part is the gene I have explained you. And this is in the diagram above, only one recombinant plasmid is shown. For simplicity, it is really lots of different ones are produced depending on the number of points at which the restriction enzyme cuts the plasmid and the other DNA. So not all the plasmids take up the gene. Few of the plasmids take up the human gene. And so how can we identify it? We can identify it using the gene marker, genetic marker. We will know what is the genetic marker later. Getting the plasmids into the vector. Now that we have made the recombinant plasmid that contains the gene that we want to insert into the recipient cell, then we have to get the plasmid into the recipient cell. In this case, it is the bacteria. The next step in the process is to get bacteria to take up the plasmids. The bacteria are treated by putting them in a solution with a high concentration of calcium ions, then cooled and given a heat shock to increase the chances of plasmid passing through the cell surface membranes. A small proportion of the bacteria, perhaps 1%, take up plasmids with the gene and are said to be transformed, that is genetically modified. The rest either takes a plasmid that have enclosed without incorporating a gene, or do not take up any plasmids at all. So So this is actually a process, and heat shock is, suddenly give heat and then stop, again suddenly give heat, again stop. Okay, so this is something to learn by heart. And only 1% of the what plasmids are taken up by the cell, and we can identify the cell again by using the genetic markers, gene markers or chemical markers. So the rest, the 99% either do not take up any plasmids at all, or have taken up plasmid that did not get the gene, because even not all plasmids get the gene, we have seen here. So it could happen that this is it has the gene, the green part, but say this plasmid do not have the gene, and a bacteria have taken up this gene. So we do not have any profit, because we want to put this gene into the bacteria, not the plasmid. So yeah, that's it. That's how you get plasmids into the bacteria or the recipient cell. Okay, so I have already told that not all the bacteria takes in the recombinant DNA. So we need to identify which bacteria takes in the recombinant DNA. I have explained how a gene is inserted along with the plasmid, so that it produces a product that lights up in ultraviolet ray. But that is a modern technique. Let's see the old technique, traditional technique. It is important to identify which bacteria have been successfully transformed so that they can be used to make the gene product. Yes, our main intention is to insert the gene into the recipient cell, so that gene is activated in that cell, and that cell can produce the product made by the gene. Because I have told many times that each proteins is highly specific to the sequence of DNA. This is done by spreading the bacteria on agar plates, each containing an antibiotic. So agar is a nutrition for the bacteria, and that agar solution contains antibiotic that kills the bacteria. So if, for example, the insulin gene had been inserted into the plasmid at a point in the gene for tetracycline resistance in PBR322, then any bacteria which had taken a plasmid with the recombinant DNA would not be able to grow on the agar containing tetracycline. So for example, all of the bacteria have resistance to that antibiotic due to a certain gene in the plasmid, and the name of the plasmid is the PBR322. There are different types of plasmids. And so suppose that the gene for insulin secretion has been inserted into the plasmid at the point that contains the gene for resistance, and so the gene for resistance is no longer in the recombinant plasmid, and the bacteria or cells or organism which have taken in the recombinant plasmid cannot produce the proteins that causes them to be resistant to the antibiotic. So they cannot grow on that agars containing the antibiotic, and we can then identify that this bacteria contains the recombinant genes. However, this technique has fallen out of favor and has largely been replaced by simpler methods of identifying transformed bacteria. We will see that later. DNA polymerase in bacteria copies the plasmids. The bacteria then divide by binary fission, so that each daughter cells has several copies of the plasmid. The bacteria transcribe the new gene and may translate it to give the required gene product as insulin. So then once we have identified the bacteria that contains the recombinant plasmid, that bacteria will multiply or divide by the process of binary fission. We will know what is binary fission, but it is simply the process in which one bacteria multiplies to many. And after it has multiplied, each daughter contains the recombinant plasmid, and so all of the bacteria will make the gene insulin, because they will make the insulin because they have the gene that codes for the protein insulin. And DNA polymerase does the copying. We know this from our chapter in A levels or O levels, mitosis and meiosis. So DNA polymerase copies the plasmids, and then the binary fission occurs, so that the bacteria cell divides and all of the daughter cell have the recombinant plasmid, and then each of the bacteria will produce the gene product which we wanted it to produce. Okay, so insulin production. Now, before this we have talked about the tools for genetic engineering. Now let's see that how will we use these tools in genetic engineering. We know that insulin is a very important hormone in our body. Okay, it converts the excess glucose in the blood into like glycogen in the cells, so that the blood glucose concentration is reduced. Okay, so one form of diabetes mellitus is caused by inability of the pancreas to produce insulin. Before insulin from genetically modified bacteria became available, people with this form of diabetes were treated with insulin extracted from pancreas of pigs or cattle. In 1970s, biotechnology companies began to work on the idea of inserting the gene for human insulin into a bacterium and then using this bacterium to make insulin. They tried several different approaches, finally succeeding in the early 1980s. This form of human insulin became available in 1983. The procedure involved in the production of insulin is shown in 19.5 figure. Okay, so what is the problem if we take the insulin from the cattle pancreas? Since we know that pancreas is the only organ that produces insulin, it's going to be costly, it's going to be time consuming, and there may be some religious factors that people do not want insulin from pigs or cattle. So but if we put the gene for insulin into a bacteria which will produce the insulin, then we can produce huge amount, like in industrial scale amount of insulin in very minimal time with minimum cost. Now let's see how does that happen. Definitely we need a vector to insert that insulin into the bacteria which will produce insulin. Now that vector, in this case the vector will be the plasmid, which is also extracted from a bacteria. Okay, and we know what vectors are. There may be different types of vectors like viruses and spears of lipoproteins, but in this case we are using plasmids from bacteria. And definitely, since pancreas is the only organ that produces insulin, and insulin is produced in the beta cells and glucagon is produced in the alpha cells, and both beta cells and alpha cells are present in the islets of Langerhans in human pancreas. So both the beta cells or alpha cells or any cells contains huge amount of genes, and it is very hard to identify the gene that produces insulin. Instead, we can easily identify the mRNA that was transcripted, so that the insulin would be produced. Okay, so the messenger RNA. So we can see here human pancreas beta cells, mRNA from beta cells, some with code for human insulin is extracted, mRNA with the code of human insulin identified and isolated, and we need to now convert this mRNA into a DNA, because we're going to be inserting the DNA. So for that we use the enzyme reverse transcriptase to reverse the process of transcription, and after reverse transcription we get the complementary DNA, and then we also add free nucleotides in the solution, so that the single stranded DNA will become a double stranded DNA by joining the complementary free nucleotides. And we need enzymes, we need DNA polymerase for producing many genes, many DNAs, because polymerase is the enzyme that does this. Okay, so yeah, we got the single stranded DNA, we're using DNA polymerase to make the double stranded DNA from the nucleotides. Yeah, so and from here we have also extracted the plasmid from the bacteria. And now Also, the DNA, the DNA must have sticky ends. Okay, if the DNA does not have sticky ends, if it has blunt ends, then the DNA will not combine with this plasmid. Okay. Then we use reverse transcriptase. The reverse transcriptase cuts the DNA of both the plasmid and the DNA for insulin, and they are cut in a way that they both have sticky ends. See, they both have sticky ends, so that the sticky ends can join. So then plasmids from bacteria are cut with the same restriction enzyme to produce sticky ends. Yes. And mRNA is taken from beta cells and used to make DNA with the help of reverse transcriptase. It is then cut to leave sticky ends. Yes. So then we use DNA ligase to join the plasmid DNA and the DNA for gene for DNA for insulin production. Like this DNA contains the genes. Okay, we used ligase enzyme. The insulin DNA and plasmid DNA are mixed together with the ligase enzyme. The sticky end bases form hydrogen bonds. The ligase joins the DNA backbone and a recombinant plasmid is produced. Good. The recombinant plasmid is introduced into the bacteria. A transformed bacteria is identified. The gene is cloned by growing the bacterium, because we need a lot of bacteria, not just few bacteria. And then finally the manufacture process. The transformed bacteria containing the insulin gene can produce insulin. These bacteria are now grown on an industrial scale in large fermenters. Insulin is extracted from the bacteria. Yes, insulin, because if this recombinant DNA is introduced into this bacteria, when in this bacteria, like transcription and translation will occur, this bacteria will also produce the insulin with the other chemicals that the bacteria produce. So we don't need the other chemicals, so the insulin has to be then extracted in the ferment. Then extraction and purification of insulin, because there are a lot of chemicals which bacteria produces, but we need the insulin. And there we have pure human insulin. See, it's very fast. Okay. Now, there are problems in locating and isolating the gene coding for human insulin from all the rest of the DNA in the human cells. Instead of cutting out the gene from the DNA in the relevant chromosome, researchers extracted mRNA from insulin from pancreatic beta cells, which are the only cells to express the insulin gene. These cells contain large quantities of mRNA for insulin that are its only source in the body. The mRNA was then incubated with reverse transcription, which comes in the group of viruses called retroviruses. As the name suggests, the enzyme reverses transcription using mRNA as a template to make single-stranded DNA. Yes, single-stranded. The single-stranded molecules were then converted into double-stranded molecules using DNA polymerase to assemble nucleotides to make the complementary strand. Yes. The genetic engineers now had insulin gene that they could insert into a plasmid to form the bacteria, to transform the bacteria E. coli. So the bacteria that produces the insulin, the recombinant bacteria or genetically modified bacteria, is E. coli. This is chosen. The main advantage of this form of insulin is that there is now reliable supply available to meet the increasing demand. Suppliers are not dependent on factors such as availability through meat trade. Genetic engineers have changed the nucleotide sequence of the genes to give molecules with different amino acid sequence. These insulin analogs have different properties. For example, they can either act faster than animal insulin used for immediately after a meal, or more slowly over a period of 84 hours to give a background blood pressure of insulin. Okay, many diabetics take both of this form of recombinant insulin at the same time. Yes, so this is very easy, and we know how we have already read here how in detail about how this happens. Okay, now I want to explain a bit something more, that this DNA polymerase does replication. So here only one or two or few single-stranded DNA will be produced, but from the free nucleotides, hydrogen bonds will be formed with the complementary DNA. It will happen without needing for any enzymes. So now we have one double strand of DNA, but we need many DNAs because we need to insert the gene into many bacteria. So we use DNA polymerase to do the process of replication and produce many DNAs. Now my question is, we then use restriction enzyme to produce sticky ends, right? Yeah. My question is, if we would produce blunt ends, what would be the problem? Okay, so here we will see this now. Yeah, so yeah, this is blunt ends, and yeah, just imagine it like this has to be this sticky on. It's pretty much easier to do it on the board. Yeah, so now we have sticky ends here. In these blunt ends, where will the plasmid join? They cannot join because look, in here, where will hydrogen bonds form? But if there are sticky ends like here and here, there in these parts there are like the nucleotide sequences, adenine, guanine, cytosine, and they have broken hydrogen bonds. So if now they're like introduced to the plasmid, they can form hydrogen bonds. There are spaces here, look, spaces here to form hydrogen bonds. But in this case, we do need the enzyme, ligase enzyme. But when it was like a single stranded, we didn't need any enzymes. So that's why it must produce sticky ends. And we use the same restriction enzyme so that like the DNA is cut at the same points at both, because if there were different points, complementary strand wouldn't have formed. And if somehow they did give blunt ends, we must add more nucleotide sequence to make it sticky end. Yeah. Okay, now let us look at other genetic markers. We know that one of the way of marking out that which one is the recombinant, which bacteria has taken in the recombinant DNA is by inserting a gene in the plasmid which is resistant to the antibiotic, to a specific type of antibiotic. And then to identify the bacteria, since the bacteria which will have taken the gene for insulin will also have the gene for antibiotic resistant, all of the bacteria will be taken to a culture of antibiotics, and the bacteria which has developed resistant towards that antibiotic will will not be affected by that antibiotic which we will use, and thus that culture of bacteria won't be dead. Okay, so but there is a problem of doing that. It could be that antibiotic resistance bacteria has evolved, and also that it could happen that they could be by mistake or by accident go out to the environment, and thus they may grow, they may also transfer that antibiotic resistance gene to other bacteria, and thus we may have a generation of genes, or we may have a group of bacteria or widespread group of bacteria that will be resistant to the penicillin, and so that disease will be untreatable. There is some concern about using antibiotic resistance genes as markers. Could the antibiotic resistance gene spread to other bacteria, producing strains of pathogenic bacteria that we could not kill with antibiotics? In insulin production, the risk is probably very small because the genetically modified bacteria are only grown in fermenters and not released in the wild. But now there are many different kinds of genetically modified bacteria around, some of which are used in situations in which their genes might be passed on to other bacteria. If these bacteria are pathogens, then we might end up with diseases that are untreatable. Well, pathogens are bacteria, we know, that cause harm to other organisms. Because of the risk of creating pathogenic antibiotic resistant bacteria, there is now less use of antibiotic resistance genes in this way, and other ways have been developed in which the successfully transformed bacteria can be identified. One method uses enzymes that produces fluorescent substance. For example, enzymes obtained from jellyfish make a protein called GFP, green fluorescent protein, that fluoresces bright green in ultraviolet light. The gene for the enzyme is inserted into the plasmid. Yes, into the plasmid. In that plasmid, also the gene for insulin production is inserted. So all that needs to be done to identify the bacteria that have taken up the plasmid is to shine ultraviolet light onto them. The ones that glow green are genetically modified ones because they have that gene which produces that enzyme, and they also have that insulin gene in them. So we need those bacteria, and we need to collect them, and we need to make them grow so that more bacteria is produced of that type. And we know that transcription will produce the mitosis will produce the same set of genes. So all of those type of bacteria will have the enzyme, the gene for insulin production. Also the gene for that produces the enzyme that gives light in ultraviolet light, but that is not a problem. We need that insulin producing gene. Okay. The same marker gene can be used in a range of organisms. Good. Another marker is the enzyme beta glucuronidase, known as GUS for short, which originates from E. coli. Okay. Any transformed cell that contains this enzyme, when incubated with some specific colorless or non-fluorescent substance, can transform them into colored or fluorescent products. This is especially useful for detecting the activity of inserted genes in plants, such as the sundew. Okay. So it is also the same thing as we have described here, but the only difference is that these enzymes change the colors of a specific type of substance, and these enzymes glows in ultraviolet ray. That's it. These are the different types of indications. Nothing much to learn there. Okay, so now we will look at promoters. Bacteria contains many different genes which make many different proteins, of course, but not all these genes are switched on at once, because we do not need every gene, every proteins at once. The bacteria make only the proteins that are required in the condition in which they are growing. For example, as we saw in chapter 16, E. coli bacteria make the enzyme beta-galactosidase only when they are growing in a medium containing lactose and no glucose available. The expression of genes such as those in the lac operon is controlled by a promoter, the region of the DNA to which RNA polymers bind at the start transcription. if we want the gene that we are going to insert into a bacterium to be expressed, then we also have to insert an appropriate promoter. When bacteria were first transformed to produce insulin, the insulin gene was inserted next to the beta-galactosidase gene, so they shared a promoter. The promoter switched on the gene when the bacterium needed to metabolize lactose. So if bacteria were growing in a medium containing lactose but no glucose, they switched both beta-galactosidase and human insulin. The promoter not only allows RNA polymerase to bind to DNA but also ensures that it recognizes which of the two DNA strands is the template strand. Within the sequence of the nucleotide is the promoter region. The promoter region is the transcription start point, the first nucleotide of the gene to be transcribed. In this way, the promoter can be said to control the expression of a gene and can ensure a high level of gene expression. In eukaryotes, various proteins known as transcription factors are also required to bind to the promoter region or to RNA polymerase before transcription can begin. Okay, so let me explain. Suppose this is a gene, not a gene, a long strand of DNA. Okay, so suppose that this part of the DNA produces one protein. Suppose this part of the DNA, which are basically sequences of nucleotides, produces another protein, and suppose that these sequences of this DNA produces another protein. So we do not need all the proteins at once. Suppose that I need a the blue protein. I do not need the red and green one to start, but when transcription will occur, they will start. So they have to know which one to start and which one not to start. That is the reason every sequence of nucleotides or genes have promoters. Okay, so once again, let's draw this gene. Suppose that, well, this is a gene, and that all of the gene will make insulin. So here at the very starting of the gene, there will be a small sequence of nucleotide called the promoter. Okay, you know the rest, called the promoter. So suppose we need to produce only insulin, so we have to also include a promoter for insulin, else the recombinant DNA in the bacteria, the process of transcription won't produce the mRNA for insulin. Thus we have to include a promoter. But okay, now let me explain you an earlier process of how promoter was used. Now promoters can be inserted into the recombinant DNA. It can be inserted into the gene for insulin, and then recombinant DNA formed with the plasmid. But earlier, when this was not possible, what happened was, well, we know that beta-galactosidase gene has a promoter, and it is activated whenever the bacteria was grown in a medium containing lactose but no glucose. So if the gene for insulin was inserted right next to the gene for beta-galactosidase, then if the bacteria was born in a medium containing lactose but not glucose, then the promoter would start transcripting that gene, and once the beta-galactosidase gene was transcripted. Since insulin is next to it, insulin is also transcripted. So when transcription occurs, that mRNA is then transcribed to make the insulin. So that was an earlier process of using promoters. Okay, so gel electrophoresis. Gel electrophoresis is a technique that is used to separate different molecules. It is used extensively in the analysis of proteins and DNA. This technique involves placing a mixture of molecules into wells cut into agarose gel and applying an electric field. The movement of charged molecules within the gel in response to the electric field depends on a number of factors. The most important are net overall charge, negatively charged molecules move towards the positive anode, and positively charged molecules move towards the negative cathode. Highly charged molecules move faster than those with less overall charge. Smaller molecules move through the gel faster than larger molecules. Composition of gel. Common gels are polyacrylamide for proteins and agarose for DNA. The size of the pores within the gel determines the speed with which proteins and fragments of DNA move. This is very simple. Let me explain you first. Okay. Just erase this part. Okay, so agarose gel, as the name says, it is a gel. So suppose that this is the agarose gel and I have cut pores in here. These are the pores. Okay. Now suppose that here I added a positive and negative charge. Okay. Now let's look here. Why is gel electrophoresis used? It is used to separate different molecules and also to analyze the proteins. We will go further into it right after I explain this. So since the molecules has to be polars, we all know protein and DNA both are polar molecules. So they will move to different anodes and cathodes at different speeds related to these factors. Okay, so for DNA is agarose solution, and for proteins the solution that we use is polyacrylamide. Alright, so electrophoresis of proteins. The charge on a protein is dependent on the ionization of the R groups on the amino acid residue. You will remember from chapter 2 that some amino acids have R groups that can be positively charged and some have R groups that can be negatively charged. Whether these R groups are charged or not depends on the pH. Okay. When proteins are separated by electrophoresis, the procedure is carried out at a constant pH using a buffer solution. Buffer solution is a solution which helps to maintain the pH of the solution constant throughout the chemical reaction. Usually proteins have a net negative charge. Okay. Gel electrophoresis. Okay, first let me explain this. Remember the amino acid, like this is the carbon, this is the carbon, and here you have the COOH acidic group. Here you have the NH2 amine group. Here you have a hydrogen. These are all constant, but this side chain, which is usually denoted by an R is what makes the amino acid. It is what on which depends the polarity of the amino acid. Will it be positive, or will it be negative, or will it be neutral? So there are about 20 kinds of R group, thus 20 kinds of amino acids. So R groups that have positively charged of NH3, and some have R groups that are negatively charged as COO minus. But usually the overall charge of the protein is negative. Now what does polar means? Polar means that the net charge of the molecule or chemical is not equals 0, but usually proteins are negatively charged, so they will be attracted by the positive anode. Okay, now let's see how we will use this technique to analyze a protein. Gel electrophoresis has been used to separate polypeptide produced by different alleles of many genes. We know what's a polypeptide. Two amino acids join together to form a dipeptide, 3 tripeptide, and 4, 5, 6, 7, 8, 9, 10 millions, thus polypeptides. And we all know what's an alleles. Alleles are different forms of a gene, like A for normal, we denote the gene for normal hemoglobin by an A, and sickle cell by an S. Okay, now let's see sickle cell anemia. For example, allozymes are variant forms of enzymes produced by different alleles of the same gene. Okay, it's something to learn by heart. There are also many variants of hemoglobin. Adult hemoglobin is composed of 4 polypeptides, 2 alpha globins and 2 beta globins. In sickle cell anemia, a variant of beta globin has an amino acid with a non-polar R group instead of one with an R group that is charged. Since one of the beta globin is non-polar, it means that beta globin won't move that much fast in an electrophoresis, gel electrophoresis of protein than the charged molecule would. We have talked about it earlier. These two variants of beta globins can be separated by electrophoresis because they have different net charges. Yes, because the normal ones are more negative and the sickle cell one is not, ones are not negative because they are non-polar. And maybe that a person may have 2 sickle cell alleles like SS, so they're not at all polar overally. But if someone has one allele which is normal, another allele which is sickle cell, then one of the alleles will move very fast, but some proteins will move very fast, and some proteins made from that allele will move very slow because that's non-polar. So yeah, we will see that in a practical. This means that hemoglobin molecules in people who have sickle cell anemia have slightly lower negative charge than normal hemoglobin, so the molecules do not move as far the gel as molecules of normal hemoglobin. Yes, the test to find out whether someone carries the sickle cell allele makes use of this difference. Figure 19.9 shows the separation of different forms of hemoglobin in samples of blood taken from the members of a family which has a child with sickle cell anemia. Okay, so let's look at this. Separation of hemoglobin by gel electrophoresis. The analysis was carried out on a family which one child has sickle cell anemia. Lane 1 contains the hemoglobin standards A, where A normal hemoglobin and S sickle cell hemoglobin. Lane 2 and 3 are hemoglobin samples from parents, and lane 4, 5 and 6 are hemoglobin samples from their children. Here we can see that the A, which are normal this is the positive anode and this is the negative, so here are these white are the samples and they will move towards the positive. But from here we can say number 1 is actually the sample which is previously known, so that you can compare. Number 2 and 3 are the parents, so we can see that in parents for both the parents half of the hemoglobins were sickle cell because they did not move far, S, it matched with the standard, and half of their hemoglobin is A, normal. So we can assume that both of the parents have genes, alleles A, S. So number 4, 5, 6. In number 4, all of the hemoglobin went to A, which means that it has AAL because it is normal, because it went to the maximum, it was attracted by the anode, the maximum, and none of them are left back, so it has normal L's, AA. But number 5, half of the hemoglobin were not that much attracted to the positive and half were attracted to the maximum. So we can assume from here that number 5 has alleles AS, but number 6 has both S, sickle cell anemia, because all of their hemoglobin was here. None of the hemoglobins moved here. It means that both the alleles produced hemoglobins with sickle cell anemia. So number 6, its genotype is SS. So this is how one of the analysis that we can do using the electrophoresis of proteins, because the proteins corresponds to the genes, because we know that the genes have been transcribed and transcribed to proteins. So yeah, this can be used as an analysis. Okay, now that we have known how electrophoresis of the protein, now let us look at the electrophoresis of the DNA. This can be used for genetic profiling or fingerprinting. Okay, let's see. DNA fragments carry a small charge thanks to the negatively charged phosphate groups. That is why they can be introduced in an electric field, positive and negative, and so they will move. And since they carry a negative charge, they will move towards the positive charge. In DNA electrophoresis, these fragments move through the gel towards the anode. Yes, just as electrophoresis of protein, the smaller the fragments, the faster they move. True. We will look at an example, the use of genetic profiling in forensic. Figure 19.10 shows how genetic profiling is carried out. Here you can see that this is a long strand of DNA. Two different restriction enzymes cut the DNA, or we can use any type of or any numbers of restriction DNA as we need. The situation will tell what we have to do. Then fragments are selected and multiplied. Well, multiplication of fragments is done by a process of polymerase chain reaction, which we will see later in this chapter. The DNA fragments are put into a gel, yes, and separated by an electric field. Okay, a radioactive probe is added so that to bind to the invisible bands of DNA, so they can blacken an X-ray film. What is this? What is a probe? Say for example, this is a fragment of DNA. This is a fragment of DNA. This is a fragment of DNA. Say for example, this fragment of DNA is adenine, cytosine and guanine. Now, what is a probe? We all know that adenine forms complementary hydrogen bonds with thymine, and cytosine with guanine, and thymine with adenine. Okay, so now since this is ACT, it will form complementary with TGA. So suppose that we have about 25 different types of probes, where CTA is one of the probe. We put the probes on these bands. First of all, definitely we need to break the double bonded DNA into a single bond by lightly heating it. So now we have free hydrogen bonds which we need to bond. So there are different types of probes, and we know what are the probes, the sequence of the probes. So, and also these probes contains a radioactive isotope so that they can be blackened in an X-ray film. So suppose that this DNA has combined with its complementary probe, which is AGT, and then once we have blackened it on the film, we can identify that AGT was the probe that has formed complementary DNA, complementary strand with the DNA. That means that this is ACT. I think I mixed up. If this is ACT, the complementary probe is TGA. So since TGA is the probe that is being blackened, then this is ACT. Common sense. So probe is actually like a marker so that you can identify which gene is present. Okay, so now let's see here. A region of DNA that is known to vary between different people is chosen because in forensic we need to compare the DNA to match. If all of the if the sequence of DNA that we have taken is same in every person, then we cannot use it for forensic purpose. Okay. These regions often contain variable number of repeated DNA sequence and are known as variable number tandem repeats, which means that the number of repeats vary. Okay, the number of DNA sequences vary, but the DNA sequences are same. Okay, only identical twins share all their VNTR sequences. Yes. DNA can be extracted from almost anything that has come from a person's body, the root of a hair, a tiny spot of blood or semen at a crime scene, or saliva where someone has drunk from a cup. Usually the quantity of DNA is increased by using the polymerase chain reaction, which makes many copies of the DNA that has been found, because we spot of blood, saliva, not that much DNA is present, right? Only very small, and we cannot see it with our eyes if we perform electrophoresis of DNA. So we need many numbers of DNA, and this is done with the process of polymerase chain reaction, which we will discuss after we have discussed the electrophoresis of DNA. Okay, we will discuss it in detail. The DNA is then chopped into pieces using restriction enzymes known to cleave it close to the VNTR regions. So like only the VNTR regions are present there, because that is what varies between person to person, unique. It is kind of like it is unique identifier for the person. So we need the VNTR parts of the DNA. Now the DNA is ready for electrophoresis. When the current is turned off, the gel contains DNA fragments that have ended up in different places. Well, we need to switch on the current first, and then the current is again turned off. So why in different places? Because not all of the charge, the sizes are the same. Yeah, these fragments are not visible straight away. No. To make the fragments visible, they are carefully transferred into an absorbent paper, which is placed on top of the gel. The paper is then heated just enough to make two strands in each DNA molecule separate from one another. Short sequence of single-stranded DNA called probes are added. They have base sequences complementary to VNTR regions. The probes also contain a radioactive phosphorus isotope, so when the paper is placed on X-ray film, the radiation emitted by the probes, which are stuck into the DNA fragments, make the film go dark. Yes, so that we can see. Now, why did we have to heat it up? Well, the DNA was double-stranded, so how can probes form complementary bonds? So that's why we needed to heat it up, so that the hydrogen bonds are broken. And we know that hydrogen bonds are easily broken in DNA with just a little bit of heat. Yes. Okay, so we end up with the pattern of DNA strands in the film matching the position that the DNA fragments reached on the agar-rose gel. Alternatively, the probes may be labeled with fluorescent stains that shows up when ultraviolet light is shown on them. Yeah, there are alternatives, and I've already explained here. See, after that we had probes. Now let's see how is fingerprinting used. Suppose that the child shares genetic material with both the mother and the possible father, showing that it is highly probable that it is the actual father. All the bands in the child will be present in the father or the mother. Look at this. Child, father. These almost matches, these almost matches, these almost matches with the child. These matches with the child. This matches with his father. This matches with his mother and slightly with his father. So these are actually the DNA, the VNTRs. You know, there are different VNTRs. So that's why many darkening area. Different VNTRs. And they are dark. They are moved from the starter culture. I mean, from the starting point according to their charge, according to their size. Yeah. So we can compare which. Like this is DNA fingerprinting, and the control DNA is actually like the DNA sequence which we know. This is used so that the experiment is much better. You will learn it in your lab. Yeah, now let us look at this one. DNA fingerprinting of a blood stain found at the crime scene of a crime compared with fingerprints from 7 subjects. This is the stain of blood at the scene of the crime, and we can see it almost, or rather fully, matches with number 3, which means that these both samples came from the same person, because VNTRs are unique to a person, which means that number 3 is the suspect, since it matches the spot of blood which we found on the crime scene. Okay, so like this, DNA fingerprinting is very useful in forensic science. Okay, so now let us learn about polymerase chain reaction. It is basically a process by which from a single DNA or a small number of DNA we can make, well, theoretically unlimited number of DNAs. Let us see how it works. The polymerase chain reaction, generally known as PCR, is used in almost every application of gene technology. It is a method for rapid production of a very large number of copies of a particular fragment of DNA. Virtually unlimited quantities of a length of DNA can be produced from the smallest quantity of DNA, even one molecule. Figure 19.13 shows the steps involved in PCR. Okay, let's see. This is the DNA extracted, and suppose this is the part of the gene, this is the part of the gene that we need to copy. Okay, so DNA is heated briefly to denature the DNA which separates the double helix. Yes, we get a single strand of DNA, because if you want to make copies of it, we need to first make single strands of DNA. Now, primer DNA added after cooling complementary base pairing. What's a primer DNA? Do you remember the term promoter? Yes, but promoter comes naturally. But when we want to artificially clone a part of the DNA, that DNA part of the DNA may not have a promoter. Maybe the promoter also involves other genes which we do not need. So artificial promoters, which are small nucleotides, small short nucleotides, are added, and these artificial promoters are called primers. So the primers act as promoter, but these are artificially made, and we can target the part of the gene in which we want to add the primer, but the promoter, it's built in. Okay. So DNA polymerase uses free nucleotides to synthesize complementary strands. Yes, we know this DNA polymerase does that. You know, we know how does this happens, the role of DNA polymerase. The gene has been copied and forms part of 2 DNA molecules. Like this, again, for each, it breaks down and another primer needs to be added, or the old primer needs to be reused again, and then again, like it keeps copying like this, and it goes on forever. Now let's see. Heating denatures the DNA, which starts a new cycle of copying, following steps 1 to 4, repeating the cycle 10 to 12 times. The genes may copy. Yes, because once again we need to heat it to break the double bonds. Again we add primers. Again polymerase does this adding with the free nucleotides present, and thus we will have more 4 strands of double the 4 double-stranded DNA. Again, it keeps going on. Now let's read. First, the DNA is denatured usually by heating it. This separates the DNA molecule into its 2 strands, leaving the bases exposed. The enzyme DNA polymerase is used to build new strands of DNA against the exposed one. However, DNA polymerase cannot just begin doing this with no guidance. A primer is used to begin the process. This is a short length of DNA, often about 20 base pairs long, that has a base pair complementary to the start part of the DNA strand. So that is to be copied. Of course, it's need to be complementary with the start part which you want, else it don't form bonds. We have already learned. The primer attaches to the start of the DNA strand, and then the DNA polymerase continues to add nucleotides all along the rest of the DNA strand. Once the DNA has been copied, the mixture is heated again, which once more separates the 2 strands in each DNA molecule, leaving them available for copying again. Once more, primer fix themselves to the start of each strand of unpaired nucleotides, and DNA polymerase makes complementary copies of them. Okay, great. The 3 stages in each round of copying need different temperatures. You need to learn by heart that. Denaturing the double-stranded DNA molecules. Denaturing the double-stranded DNA molecules to make single strand once requires a high temperature of around 95 degrees Celsius. Okay. Attaching the primers to the end of the single-stranded DNA, known as annealing, requires a temperature of about 65 degrees Celsius. Okay. Building up complete new DNA strands using DNA polymerase requires temperature from 72 degrees Celsius. The DNA polymerase used for this process comes from the microorganism that has evolved to live in hot temperatures, hot environments. Most laboratories that work with DNA have a machine that automatically changes the temperature of the mixture. Yes. The DNA sample is placed into a tube together with primers, free nucleotides, a buffer solution, and the DNA polymerase. Why buffer solution? To keep the pH of the solution constant. The PCR machine is switched on and left to work. The tubes are very small and a very thin wall, so the temperature in the machine changes. The temperature inside the tube changes very quickly because thin walls means transfer of heat is faster, either out of the system or into the system. Yeah. You can see that theoretically this could go on forever, making more and more copies of what might originally have been just a tiny number of DNA molecules. A single DNA molecule can be used to produce literally billions of copies of itself. Just a few hours, PCR has made it possible to get enough DNA from a tiny sample, for example, a microscopic portion of a drop of blood at a crime scene. Tech polymerase, so the first heat-stable DNA polymerase to be used in PCR. It was isolated from thermophilic bacterium Thermus aquaticus, which is found in hot springs in the Yellowstone Park in the USA. It is valuable for PCR. For 2 reasons. First, it is not destroyed by the denaturing process, so it does not have to be replaced during its cycle, because denaturing is the highest temperature, and so the polymerase may be denatured also. So we need to add more polymerase to it, but we do not need the polymerase because that is heat stable. It can work again. Second, its optimum temperature means the temperature for the elongation process does not have to drop below that of the annealing process, so efficiency is maximized. So this is the annealing process, and so we do not need to then this is the elongation process. So once it's in the annealing process, in the elongation process, it don't get denatured. Look, so efficiency is maximized because that elongation process can take at any temperature, higher temperature than the annihilation process, which means that we do not have to just always change it. So yeah, efficiency is maximized, so is profit. PCR is now routinely used in forensic science to amplify DNA from the smallest tissue sample left at the scene of crime. Many crimes have been solved with the help of PCR together with analysis of DNA using gel electrophoresis. We use analyzing of DNA for DNA fingerprinting, you remember, right? Okay, now let us study microarrays. Microarrays have proved a valuable tool to identify the genes present in a organism's genome and to find out which genes are expressed within the cells. They have allowed researchers to study very large numbers of genes in a short period of time, increasing the information available. A microarray is a small piece of glass or plastic, usually 2 cm square. Short length of single-stranded DNA are attached to this support in a regular 2D pattern and 1/10,000 or more different positions per centimeter square. Each individual position has multiple copies of the same DNA probe. It is possible to search database to find DNA probes for a huge range of genes. Having selected the gene probes required, an automated process applies those probes to the positions on the microarray. Okay, let me just draw. Suppose that, well, this is the tray and we have holes here. It's a horrible drawing, but yeah. And in these holes, say for example, here we have one type of probe, and here we have another type of probe. Here we have another type of probe. Whoa, sorry, just erased it. Yeah, so say for example, here we have another type of probe. Here we have another sequence of probe, to be exact. And there we have another sequence of probe. Say for example, this is one sequence of probe, maybe A, G, T, C, A. This is another sequence of probe. This is another sequence of probe. Each color different sequence of probe. And we use many of these probes. Like in the red, it's not gonna be only one probe. It's gonna be like many probes. So, and we know which hole is which probe. So that's a microarray. See? Okay, now let's see here. When microarrays are used to analyze genomic DNAs, the probes are from known locations across the chromosome of the organism involved and are 500 or more base pairs in length. Okay, the probes are very long, good. A single microarray can even hold probes from the entire human genome. Good. Microarrays can be used to compare the genes present in two different species. Suppose we have two different species and we have collected their genes, and then we put the genes in the probes. First of all, species 1, and then we see that only red and green takes in those genes, and then we have species 2, we see that green and blue. So, and we know the sequence of the probe, so we know the sequence of the genes of those species. So yeah, but you cannot see it with your eye. So the DNA needs to be labeled and the single-stranded DNA needs to be formed, because a double-stranded DNA won't form complementary base with the probes. So yeah, let's see how this happens. The DNA is labeled with fluorescent tags, so that for example DNA from one species may be labeled with green tags and DNA from the other species labeled with red tags. The labeled DNA samples are mixed together and allowed to hybridize with the probes on the arrows. And they can hybridize because it will be formed into single-stranded first. We will look at that. Any DNA that does not bind to probe or the micro is washed off. It means that the probe is not complementary to none of the probe is complementary to those DNAs. The microarray is then inspected using ultraviolet ray, which causes the tags to fluorescent. When this happens, we know that hybridization takes place because the DNA fragments are complementary to the probes. Green and red fluorescent spots indicate where DNA from one species has hybridized with the probes, where the DNA from both species with the probe. A yellow color is seen because red and green mix together to give yellow. So suppose in one probe, say in orange probe, both the species DNA has hybridized. So it will give yellow, and so we will understand that the sequence of the probe, which is orange, we will know that from our computer, and then we have the knowledge of complementary DNA, and so we know that this nucleotide sequence of nucleotides is present in both the species. And if suppose only orange has only red fluorescence, it means that only species number 1 has hybridization. It means that only species from genes from species 1 have hybridized, which means that the probe, say the sequence of the probe of orange is AG, and so we will know that the sequence of the gene in the species is cytosine and thymine. Thymine and cytosine, because AG complements with A with thymine and G with cytosine. Yeah, it's that easy. And why are we using fluorescence or using like a label tag? So that it lights up, because we cannot see the DNA with our eyes. Lights up in ultraviolet rays. Green and red fluorescent spot indicates that from one species has only hybridized, so DNA from both species hybridized with the probe by yellow color is seen. Yellow spots indicate that the two species have DNA with exactly the same base sequence. Okay, this suggests they have the same genes. If there is no color for a particular position on the micro, it means that no DNA has hybridized with the probe and that a particular gene is not present in either species. Yes, it's a matter of common sense. The micro is then scanned so that the data can be read by the computer. Data stored by the computer indicate which genes are present in both species, which genes are only found in one species, and which genes are not present in either species. So this is a very easy way to compare the genes, much more easier than electrophoresis, because in electrophoresis only not that much genes can be compared. But if you do it in a microarray, we already have read that one microarray may contain the genome of a whole human. Wow. Now, okay, good. Now let's see another example. Microarrays also make it possible to detect which genes are being expressed at any specific time in each cell in the body. For example, the genes that are expressed in a cancer cells are different from those active in non-cancer cells. Microarrays are used to compare which genes are active by identifying genes that are being transcribed into mRNA. Okay, this should be transcribed into mRNA. Whatever. Obviously, which genes are active, those genes will be used to produce the proteins. And if those genes are active, then mRNA for those genes will be produced. So if you collect the mRNA and then use reverse transcription to produce the genes, the complementary DNA strand, and then that single-stranded complementary DNA strand used in the DNA probe, and we see that those genes are active, then we can understand that which genes are active. We can also understand the level of activity, because more mRNA being produced means that in ultraviolet ray it will give more fluorescence, because deeper colors, because many mRNAs, if there are 2 mRNAs only which are tagged, of course, then only a few light. But if there are 1,000 mRNAs which are again also tagged, then there will be deeper light. That's a common sense. We can understand which genes, we can compare genes, we can understand which genes are active, we can also understand the level of activity of each genes. Okay, so the mRNA from 2 types of cells is collected and reverse transcriptase is used to convert mRNA to complementary DNA. As the quantity of mRNA in a cell at one time is quite small, the quantity of complementary DNA need to be increased by PCR. Okay, if it's small, we can increase by PCR, but we will not increase it by PCR if we are examining the level of activity. Okay, the complementary DNA is labeled with fluorescent tags, denatured to give single-stranded DNA. Definitely, the complementary DNA is, when reverse transcription occurs, the mRNA and complementary DNA which will be formed will produce a double strand, because mRNA, free nucleotides, another complementary strand makes a whole DNA. But we do not need the double strand, we need only single stranded, the complementary DNA part. Okay. To to give single-stranded DNA and allow to hybridizate. The problem there is spots on the iris that fluorescent indicate the genes that were being transcribed in the cell. The intensity of the light emitted by each spot indicates the level of activity of each gene. A high intensity indicates that many mRNA molecules are present in the sample, while a low intensity indicates that there were very few. The results therefore not only show which genes are active, but their level of activity. This information is changing the way in which cancers are treated. Let me explain why did we have to denature it. Suppose that, well, this was the DNA, this is the mRNA, and in the mRNA, let me just denote it with M, where there will be, we need to reverse transcriptase, then another new strand. This is the new strand, which is the original strand that was copied, but all of it made a double-stranded. With the mRNA, it made the double-stranded DNA. We do not need it, so we need this only. We only need the new strand, which is formed by reverse transcription, which is complementary to the strand of the DNA that was copied. Now, so what we do? We denature it. We take only the strand, which is the original strand that was copied, and then we use it in microarrays. Okay, so bioinformatics, research into the genes that are present in different organisms and the genes that are expressed at any one time in an organism's life, generates huge quantities of data. As we have seen, one DNA chip alone may give 10,000 pieces of information about the presence and absence of genes in genomes or the activity of genes within the cells. Gene sequences has also generated huge quantities of data. This technique establishes the sequence of base pairs in sections of DNA. Sequencing DNA is now a fully automated process, and the genomes of many species have been published. There are also vast quantity of data about the primary structure, shapes, and functions of proteins. Bioinformatics combines biological data with computer technology and statistics. Okay, so there are vast amount of data that has been collected. Now, analyzing, processing, and collecting the information is the work of a bioinformatician who study bioinformatics. So basically the information is so big. I mean, the area of analyzing, collecting, and also processing the biological information and data is so large that a new sector had to be formed, which is bioinformatics. So nothing much to learn from here or explain from here. You can learn a lot, but it is hard to nothing to explain actually. Look, it builds up databases and allows links to be made between them. The databases hold gene sequences of complete genomes, amino acid sequences of proteins and protein structures. Computer technology facilitates the collection and analysis of this mass of information and allows access to it via the internet. There are databases that specialize in holding different types of information. For example, on DNA sequences and the primary structure of proteins, the information needs to be in a form that can be searched. So software developers play an important role in developing systems that allow this. In 2014, these databases held over 6 × 10⁻11 base pairs or 600 GBP, equivalent to 200 human genome equivalents or huges. A huge is 3 × 10⁻9 base pair. Database that holds the coordinates requires to show 3D model, hold details of over 100,000 different proteins and nucleic acids. The quantity of data is vast and growing at an exponential rate. It's so big. The database holds data on the genomes of eukaryotic organisms. Among others, it holds the human genome and the genomes of zebrafish and mice that are used in a great deal in research. UniProt, Universal Protein Source, holds information on the primary sequence of proteins and the functions of many proteins such as enzymes. The search tool BLAST is an algorithm for comparing primary biological sequence information, such as the primary sequence of different proteins or the nucleotide sequences of genes. Researchers use BLAST to find similarities between sequences that they are studying and those already saved in databases. When a genome has been sequenced, comparisons can be made with other genomes. For example, the human genome can be compared to the genomes of the fruit fly, the nematode worm, or the malarial parasite. Sequences can be matched and degrees of similarities checked. Okay, we can also check how much similarity there. Similarly, comparisons can be made between amino sequence of proteins or structured proteins. Close similarities indicate recent common ancestry. Yes, human genes, such as those that are concerned with development, may be found in other organisms, such as Drosophila. This makes Drosophila a useful model for investigating the way in which such genes have their effect. One thing let me explain. Why does close similarities in protein structures indicate common ancestry? Well, evolution occurs gradually. Gradually the genes are being changed and thus the structure of the proteins are being changed. So if there is a similarity between the structure of proteins between species, it means that they may have been evolved from the same ancestors. Okay, like one organism may be evolved into many different types of other species. Okay, we'll learn this in another chapter. Okay, microarrays can be used to find out when and where genes are expressed during the development of a fruit fly. Researchers can then access information about these genes and the proteins that they code for. For example, they can search database for identical or similar base sequences in other organisms, compare primary structures of proteins, and visualize the 3D structure of proteins. Okay, Habditis elegans was the first multicellular organism to have a genome fully sequenced. It had fewer than 1,000 cells in its body and about 300 nerve cells. It is conveniently transparent, allowing the development of fate of each of its cells to be mapped. Because of its simplicity, it used as a model organism for studying the genetics of organ development, the development of neurons into a nervous system, and many other areas of biology such as cell death, aging, and behavior. Okay, all information we have about genome of Plasmid is now available in databases. This information is being used to find new methods to control parasites. For example, being able to read gene sequence is providing valuable information in development of vaccines for malaria. So, researchers in the field of microbiology, biochemistry, molecular biology needs this information. But before that, this information needs to be collected, processed, and analyzed. Okay, so this is done by the bioinformatician. So bioinformatics is the collection, processing, and analysis of biological information and data using computer software. Nothing to explain here actually.